morning, students. Today is the fourth part of chapter Principle of Inheritance and Variation, plus 2 class biology. So, in the previous part 3, we have discussed about the terminologies or the definitions of the terms that are used, that will be used in the genetics. So, let's start. Let's start with our first topic, today topic. In the previous part 1, 2, we have covered about the Mendel and the work of the Mendel, and Mendel crossed on a specific plant known as pea plant, Piscium sativum. So why Mendel selected a pea plant, or what is the advantage? What is the advantage of having a pea plant? So pea varieties are easily available. First advantage, pea plants are easy to cultivate. They can be easily cultivated anywhere, and the life cycle of pea plant is for few months, near about 3 months. Means pea plant will show result in 3 months only. So we can get result very early and very easily. So if we have to maintain the cross with 3 to 4 generation, so we can easily get the result in near about 2 years. So it is very advantageous. Contrasting characters are available. Contrasting traits means tall, dwarf, color, position, shape. So contrasting traits are easily observed. Flowers are bisexual, and flower has the capacity for self-pollination also. Flower can be cross-pollinated manually with the human efforts, and hybrid fertiles are produced. Means the hybrids that are produced they are fertile. So these are some advantage of selecting the pea plant. First, pea variety are easily available, easy to cultivate. Lifespan is for few months. Contrasting characters are available: bisexual, self-pollinated, cross-pollination, and fertile. Yes. Again, these are some 7 characters that was discussed in slide 2. So Mendel selected these 7 characters: height, shape, colors, coat color, pod shape, pod color, pod position. And in the height, tall and dwarf; in the shape, rounded, wrinkle; in the color, yellow, green; in the seed coat color, green, white; in the pod shape, green, full and constricted; pod color, green; flower position, axial and terminal. So these are some 7 contrasting characters that are used for Mendel experiment. Yes. Now Mendel's law of heredity. This is very important topic for your class board exams. Now, first is the paired factor. The factors that are responsible for the transmission of character. Means factors are responsible for transmission of character. These factors are transferred from one generation to next generation, and according to this principle of paired factor, these factors are present in pair. These factors are present in pair. So first principle is very easy. Principle of paired factor means capital T, capital T, capital T, small t, small t, small t. These are some paired factors. Then law of dominance. This is also very easy law. Just we have to remember, or we have to write down that according to law of dominance, one allele is dominate over other. One allele is dominate over other means one allele will express its character and the other allele will not express its character. So the allele which express themselves are known as dominant alleles, and the alleles which do not express their character are known as recessive alleles. So law of dominance: in hybrid dominant character suppresses the expression of recessive character. Means dominant character will suppress the character of recessive one. And next, Mendel law of segregation. uh, it is very easy law. This law states that when a factor is present in pair, or when a pair of factor are present and they are bring in close in the hybrid, then these will segregate. Law of segregation. Segregation means separate. So we write down simply, this law state that when the pair of factor are present together, then this pair of character will separate during the formation of gamete. So this is a simply you have to mention here when a pair of factor are present, these pair of factor will segregate during the formation of gamete. Yes, inheritance of one gene. One gene means single gene is taken during the study of inheritance pattern. Single gene means height character, or these cross are known as monohybrid cross. These cross means one gene cross are termed as monohybrid cross. So the cross between the two parents of differing in only one pair of contrasting character means when we choose a height as a character, when we choose height as a character, then we maintain a cross with height, that is tall plant is crossed with dwarf plant, and we obtain the ratio, that is ratio 3:1. So the ratio 3:1 is the monohybrid ratio, and the cross is known as monohybrid cross. Hope you understand this. Mono means single. So when single characters are choose, and this single character will use for crossing. Single character means height is a character. So when the height taken as a character, that is tall and dwarf, is crossed, this is known as monohybrid cross. Yes, this is monohybrid cross, already explained, but let's have a quick revision. Yes, in this single character is selected, that is height is selected, and here capital T tall height, here small t dwarf height is there, and these two parent plant P1 and plant P2, or parent P1 or parent P2 will produce two type of gamete. Capital T will form single type of gamete capital T. Small t will form single type of gamete known as small t, and this capital T and small t will combine to form first generation hybrid known as capital T and small t, and this first generation will again crossed. This first generation will again crossed capital T small t capital T small t, means for finding the second generation, for finding the second generation, first generation will be crossed, self. Now I'm again repeating, for obtaining the second generation, first generation will be self cross. Here f1 is written, f1, f2 is obtained by crossing f1 and f1. So capital T small t and capital T small t will again crossed to obtain 3:1 as its phenotype, and 1:2:1 as its genotype. Yes, again this is given here, monohybrid cross. This is parent tall, this is parent dwarf, first generation hybrid, capital T small t, all plants are tall, and in second generation, capital T here, small t here, and capital T here, small t here, and these are crossed, this capital T, and in lower box capital T, small t, capital T here, and small t, and small t. This is the way to obtain the phenotype and genotype ratio, and the phenotype ratio is 3:1 and the genotype ratio is 1:2:1. So this is all about. Thank you.